In the late 20th century, botanists confronted a nightmare disguised as a blossom, corpse flowers that smell like rotting meat, yet are scientifically exquisite, not monstrous legends. Amorphophallus titanum, documented since the 1870s, reached Western greenhouses again between 1950 and 2000, astonishing researchers with lifespans, enormous size, and unsettling olfactory mimicry. Chemical analyses from the 1970s onward revealed a calculated cocktail: dimethyl trisulfide, putrescine, cadaverine, compounds identical to decomposing flesh, carefully released to deceive hungry insects. Using thermocouples in the 1990s, scientists confirmed the bloom heats itself, reaching mammalian temperatures so the reeking vapor rises convincingly, like breath from a carcass. The plant's metabolism surges briefly at night, synchronized with local carrion beetles' activity, an evolutionary trick documented through nocturnal field observations in Indonesian rainforests during late 20th century expeditions. Despite decades of vegetative growth, the colossal bloom lasts just days. Botanical gardens worldwide carefully logged these rare events, treating each emergence like an unpredictable eclipse. Between 1980 and 2000, each documented corpse flower bloom drew thousands, forming lines around conservatories, where scientists balanced public fascination with controlled, meticulous data collection. Far from greenhouses, parasitic Rafflesia arnoldii, known from colonial-era records, re-emerged in 20th century field studies, a petalless giant embedded directly in jungle vines' tissues. 20th century botanists confirmed Rafflesia lacks leaves, stems, and roots, existing almost entirely as microscopic threads within its host, surfacing only briefly as a massive flower. Careful monitoring in the 1960s through 1990s showed Rafflesia buds developing for many months unseen, then decaying rapidly, leaving only soft, blackened tissue on the forest floor. Late 20th century molecular work revealed Rafflesia possesses drastically reduced chloroplast genomes, stripped of photosynthetic machinery, confirming its total dependence on hosts for energy and metabolites. Genetic comparisons in the 1990s uncovered horizontal gene transfer between Rafflesia and its host vines, documented sections of DNA apparently stolen, blurring boundaries between parasite and victim. Corpse flowers and Rafflesia evolved separately, yet both converged on the same strategy: mimic carrion precisely enough that flies and beetles mistake them for feeding grounds. Behavioral experiments from the 1970s onward showed scavenger insects landing, probing, and laying eggs on these flowers, responding as though encountering actual carcasses rather than elaborate botanical illusions. Beyond Rafflesia, botanists cataloged numerous parasitic plants between 1950 and 2000, from dodder vines to broomrapes, each siphoning nutrients from unsuspecting hosts through intimate vascular connections. Microscopic imaging during these decades detailed haustoria, specialized invasive organs piercing host tissues, fusing with transport vessels, turning other plants into unwilling life support systems for parasites. By the 1990s, controlled experiments revealed parasitic plants manipulate host hormones and signaling molecules, altering growth patterns, documented as twisted stems, stunted leaves, and distorted flowering times. Comparative anatomy across decades showed many parasites abandoning unnecessary organs, leaves shrinking, roots fading, evidence of evolution trimming everything except the machinery for invasion and reproduction. Radioisotope tracing in mid-20th century studies followed labeled nutrients flowing from host to parasite, glowing images confirming direct theft rather than any mutualistic exchange of resources. While corpse flowers mimic decay, forensic research from the 1950s onward charted real decomposition, cataloging insect succession and microbial blooms that transform a body into fleeting habitat. Chemical profiling of decomposing tissue identified overlapping compounds with corpse flower scents, confirming these plants evolved to copy the molecular language of death extremely precisely. Entomologists observed how carrion insects inadvertently disperse pollen, their bodies dusted as they search for food or breeding sites, unwitting couriers in this morbid communication network. Newspaper archives from 1950 to 2000 reveal headlines combining horror and awe, framing these blooms as botanical freak shows, while scientists quietly compiled meticulous phenological records. In the same decades, agricultural reports documented parasitic plants devastating crops, reducing yields and prompting international collaborations to understand their biology, not merely eradicate apparent botanical villains. By century's end, corpse flowers and parasitic plants served as emblems in museum exhibits, illustrating extreme evolutionary pathways rather than simple curiosities or botanical abominations. During the mid-20th century, expanding research funding, partly driven by Cold War competition, indirectly supported deep dives into plant physiology, parasitism, and bizarre reproductive strategies like corpse flowers. Field botanists, often operating on limited budgets, logged precise coordinates, measurements, and photographs whenever they encountered Rafflesia or related parasites, building irreplaceable 20th century baselines for future comparisons. Advances in microscopy and staining techniques across 1952,000 turned invisible parasitic filaments into vivid images, confirming long-standing hypotheses about how deeply they infiltrate host tissues. Early molecular phylogenies placed corpse flowers and parasites across the angiosperm tree, demonstrating that extreme deception and parasitism arose multiple times, not from a single monstrous lineage. Ecologists during this period emphasized that deception ultimately supports new life. Flies fooled by flowers still reproduce, while real carrion nourishes larvae, microbes, and scavenging vertebrates. Surveys in tropical forests revealed networks of parasitic plants, each tapping trees, shrubs, or vines, collectively rerouting energy flows in ways only partly understood by 20th century researchers. By 2000, documented gene transfer, shared sense, and interwoven anatomies forced biologists to reconsider strict separations between organisms, hosts, and the environments they chemically manipulate. Archival interviews captured scientists describing these organisms not as abominations, but as elegant outcomes of selection, perfectly adapted to niches involving death, decay, and exploitation. Corpse flowers and parasites demonstrate that evolution can sculpt organisms to read and broadcast signals of decomposition, turning the end of one life into opportunity for another. Every unsettling detail here, stench chemistry, parasitic threads, stolen genes, emerges from published studies, field notes, and photographs, reminding us reality often exceeds invented horror. If documented oddities like these fascinate you, subscribe for more strange but true explorations into real biology that feels disturbingly close to science fiction.